Hi, everybody. We're a few minutes away from the start of the webinar. We're going to start in a few minutes, but I just wanted to say hello to everyone and thank you for joining us. We're going to have over 100 people on today's webinar. As we are starting and preparing today, I wanted to give everyone the chance to continue to learn more about the Autism BrainNet by going to www.takesbrains.org/sign up. You will get a quarterly newsletter and updates about things like this, the webinar. You may have heard about the webinar through being an Autism BrainNet registrant. But we're going to go ahead and start in a few minutes. And just hang on a few seconds. I know we've got about 18 people on the line right now. Okay, guys, we're going to go ahead and get started. It's 1 minute after 12, but I wanted to make sure everybody has as much time as they need to ask questions at the end. So I wanted to go ahead and get started. My name is Alicia Halliday. I'm the Chief Science Officer of the Autism Science Foundation. The Autism Science Foundation works very closely with the Autism BrainNet to ensure that everyone knows the options about brain tissue donation, both families who are affected with autism and those who are not affected with autism. Today, we are very honored to have Dr. Neelroop Parkashak, who's now at UCSF. It's truly an honor to have him to discuss his research today. He has authored many research publications in the understanding of the biological basis of autism spectrum disorders. He uses both blood and postmortem brain tissue. And we asked him today to kind of give a little bit of a summary of what the projects he's been involved in and what he's done. There is lots here, so put your seatbelts on. I have to say, Dr. Parkashak is one of those rare gems with an amazing background and a fabulous training who really wants to contribute as much as he can. And he's the very reason why training grants for autism are offered, is to nurture investigators like Dr. Parkashak. So we're very thankful he's joining us. He was a graduate of Rice University in Texas, and then he moved to UCLA and got his PhD in neuroscience. Because that just wasn't enough to get a PhD, he went then on to get an MD as part of a joint PhD-MD program at UCLA. In addition to his research experience, he's worked at the Department of Neurosurgery at UCLA and is now a resident at UCSF. All of this was done while working incredibly hard trying to understand why the genetics of autism has been so elusive. He's published 15 research articles in major journals exploring the genetics behind autism. He and his mentor, Dr. Dan Geschwind at UCLA, have been pioneers in demonstrating through scientific research that autism is not just single genes working separately, but rather the interactive network of different genes working together at once. He's lent his expertise not only to understanding the genetics of autism, but how they contribute to sex differences and potentially through epigenetics, which may explain how genetics and environmental factors interact. Again, as this is an Autism BrainNet webinar, we hope you're considering registering while you're in front of a computer. It's www.takesbrains.org, sign up. Dr. Parkashak has plenty to discuss, so before I turn it over to him, I want to talk about a few housekeeping notes. First of all, you're all on mute. You can yell at your dog or yell at your kids or go to the bathroom. This means, though, that you can't ask questions via the phone. So instead, if you want to ask a question, which we will answer at the end of the webinar, you can use the text box in the GoToWebinar panel on your upper right-hand screen. You can also email it to me, and there's my email address, ahalliday@autismsciencefoundation.org. And if you're having problems seeing the GoToMeeting screen or GoToWebinar screen, go ahead and close other windows, log out of other programs, or click, it's actually a yellow flower on your control panel. And this is what the GoToWebinar control panel looks like. If you go to this box right here, the chat box, you can type in messages. And then if you lose the window, you can hit this particular kind of orange flower. That will take you back to the GoToWebinar if other programs are closed. Okay. So we have about 35 people on. Without any further ado, what I'm going to do is I'm going to turn it over to Dr. Parkashak. I'm going to change the presenter and make him the presenter so that he can start his presentation.
Hi, everybody. Thank you for joining us this afternoon. I'm going to be talking to you about, and thank you to Alicia for the really nice introduction. I won't tell you anything else about myself other than I usually go by Neil. So today we're going to talk about understanding typical brain development and autism through genetics and epigenetics. And as it was already mentioned, I did this work in Dan Geschwind's lab over the course of 5 years, plus or minus, well, plus really 2 years, as things kind of carried over while I was in my MD training the last 2 years. So let's get into the meat of things. Please ask questions. There will be some nitty-gritty biology and and bioinformatics discussed, but I hope that I'm able to convey the bigger picture and the importance of postmortem tissue to a lot of the work we've done in understanding the role of molecular biology and understanding autism. So the autism spectrum is encompassed by what we call autism spectrum disorder, and there are two major phenotypes that define autism. There are deficits in social communication and restrictive and repetitive behaviors. Autism is very prevalent, 1 in 68 individuals in the US, and this estimate has actually remained stable for the last few iterations that the CDC has released. And autism is diagnosed based on symptoms before age 3, which is why we call it a neurodevelopmental disorder. 83% of individuals have significant social, occupational, or other life impairments, which we call, although the way we kind of speak of that medically is comorbidities. So individuals with autism frequently have intellectual disability of some form or another, epilepsy, attention deficit hyperactivity disorder, language impairments, and other comorbidities, which I won't go into. But what this means for the study of autism is it's very difficult to distill what is contributing to autism, since there's all these other factors that are involved in any given individual who has autism. And the one thing with defining autism is that there's this sort of gray area where we say they have autism unless there is another disorder that better explains the deficits. So, for example, if someone has severe intellectual disability, that would be usually defined as an IQ less than 40, based on typical neuropsychiatric testing, then they often do not get a diagnosis of autism. However, there is some gray area there. Interestingly, in the overall autism spectrum, about 54% of individuals have an IQ less than 85. So that means that a good fraction of individuals with autism have IQ in the normal or higher than normal range too. So one of the big changes in the way we think about autism over the last 2 decades has been the acknowledgement that genetics contributes to the risk for autism, and that's been largely supported. Yes.
Neil, I hate to interrupt you, but I don't know if anyone else is having this problems. I'm not seeing your slides advance.
Oh, you're not?
I don't. Yeah. So, are you seeing them in normal view or in PowerPoint view?
I see them in PowerPoint view, and they do seem to be advancing. Are other people having the same issue?
Can people write in the text box if they're having, if other people are having issues? Yeah, click on that. Maybe just take it out of PowerPoint view and keep it in normal view and then advance it that way.
Sure.
Yeah, then I saw it. I just saw the next one. So yeah, maybe just keep it like that.
Okay. Let me see what I can do with that, because yeah, that might influence how some of the slides appear. Because I won't be able to use the animations the same way, but we'll work with it.
Okay, so, thank you. I don't know if other people are having the same problems. Can you make a note in the chat box?
Okay. Shall I continue for now?
Yes, I'm sorry. Yes, please.
Okay. So right. So I won't have the animation. So sometimes some of these slides might seem very busy initially. I apologize for that in advance. This way, though, I can highlight some of the points, I guess. So here, the thing is that we know now that monozygotic twins are highly concordant for a diagnosis of autism, meaning that if one twin in an identical twin pair has autism, the other twin is 36 to 95% chance of having autism. And in many of the studies I've seen, it's more like 70 or 80%, not in the lower 36% range. And for dizygotic twins, meaning non-identical twins, the concordance rate is much lower, in some studies as low as 0%, and in other studies as high as 31%. So this suggests that when you share a genome with someone, you have a higher risk of autism. Another observation supporting that is that when one child has autism, it increases the chance of the second child having autism to 15%. So the prevalence in the general population is between 1 to 2%. It increases to 15% when it's your sibling. So these observations together tell us that genetics can offer clues to the cause of autism, and that has led to many studies. These studies look at the genetics of autism. So the thing about the genome, the human genome, is it varies across individuals, and these mutations contribute to genetic diversity. So there are several types of variants in the genome, and you have to take this in the context of we have about 3 billion base pairs in our genome, and out of these, about 3.7 times 10 to the 6, so that's 3,700,000 of the base pairs out of 3 billion are affected by single nucleotide variants. These are one DNA base pair change in the genome. The other thing that contributes in human genetic variation is insertion deletion variants. So these are situations where you insert one or a few base pairs or remove them entirely. And there's about 36,000 of those per individual. 360,000, sorry. And then you have copy number variants, and these are large swaths of the genome which are inserted or deleted. They can be anywhere from 100 to up to 1 million base pairs in length, and there's about 743 of these events on average per person in the general population. So what does that mean for our understanding of autism? Well, when we're studying the genetics of autism, it's very difficult to distill risk from chance variation. There are many different studies that aim to understand what's called the genetic architecture of autism, and that has to do with how each of these different variants in different ways contributes to autism risk. So far, many variants have been confirmed as contributing to autism, but thousands of variants are predicted to contribute, and these variants are going to be distributed across hundreds of genes. So autism, the genetics of autism, the genetic architecture of autism is going to be a complex architecture. It's not going to be simple like certain disorders or diseases we know about in the general population, like for example, cystic fibrosis is a simpler genetic architecture. So genetic variation is, of course, not the end point. It's just the starting point, and it affects multiple levels of biology. So mutations by themselves provide little information about what will happen in the brain. So here you have a mutation occurring in the genome, and what we observe is what's happening in various, in the clinic, for example, we see what's happening in brain regions by doing an MRI on someone. We can see what's happening cognitively by either neuropsychological testing or seeing their academic performance. And we can see what's happening behaviorally based on interacting with the individual, whether it's in the clinic or in a day-to-day life. But there's a lot of things that happen between a mutation in the genome and a manifestation as a cognition or behavior. And one of the areas that's kind of higher resolution than looking at the whole brain is looking at microcircuits and long-range circuits. So these are the connections within and between brain regions. And we can study these less so in human. We have certain imaging modalities which let us study them to a certain extent, but they're mostly well understood in model organisms like mouse. And then what's happening in these circuits is mediated by cell types and cellular subtypes. So these include neurons and glia, and these are the cells that compose the entire brain. And then what's happening in these cells is determined by what's happening in the cellular subcompartments. So the cellular subcompartments include the nucleus of the cell, which is the place where sort of all the genome is converted to different other molecules, and then where signaling occurs, which is the synapse. And that's how the cells talk to each other. So basically, when there's a mutation in the genome, it's somehow affecting molecules in each of these cellular subcompartments, and that's having some effect up into the behavioral level. But this isn't all. These levels also vary across development. So from fetal brain to adult brain, there's a change in how these cell types are organized, how they're talking to each other, and how they're structured in these circuits. And so what happens in the genome happens from early on in from basically conception, and so it's influencing all of these levels of biology across time, and that adds another layer of complexity. So in order to understand these levels of complexity in human brain and actually in other model organisms too, we look at molecular biology, and molecular biology has several levels of investigation. One of them is at the RNA level, which we call the transcriptome. So these are RNA transcripts. They include mRNA, messenger RNAs, LNC RNA, which is long non-coding RNAs, and miRNA, which are microRNAs. And these RNAs are transcribed into proteins. So they form what we call the proteome. And proteins really make the structure of the cell. So they make all the molecules that talk to each other and mediate signaling that is basically how messages from the environment enter the cell and influence the biology. And then these proteins in turn can bind to RNA and can also bind to DNA. And that has to do with what's going on in the nucleus. So the nucleus contains the DNA, but the DNA is modified by proteins. And these proteins, in combination with the DNA, make what we call the epigenome, and that has to do with chromatin. So chromatin is how certain proteins are bound to and wind up the DNA, and the DNA is opened up when certain signals are received inside the nucleus. So the important message from all of this is that everything between DNA, which we call the genotype, and what we observe, which is the phenotype, is epigenetic. So the RNA, the way the chromatin is structured, and even the way the proteins influence the RNA and the DNA can be considered epigenetic, because any environmental stimulus can basically initiate a cascade of signaling into the cell and change how these levels of molecular biology function. So the premise in our lab is that given that autism is a set of shared phenotypes based on what I talked to you about in the initial slide, we believe that there's some shared molecular neurobiology. That means there might be some shared signaling pathways at either the transcriptome or the epigenome or the proteome, or there might be some shared cell types or circuit levels that are affected in the disorder. So here, this slide would be a little easier to understand with animations, but I'll start from this side. So basically, I want to discuss the role of human postmortem tissue studies in autism. And the key thing to know is that humans are very different from mice, zebrafish, worms, other model organisms that are commonly used in neuroscience. And I'll just highlight the differences between humans and mice. So the human brain is very large, first of all. If you were to look at this mouse brain, this is of course not to scale. The mouse brain would just kind of fit in barely in this kind of bottom part of the human brain stem here, and not even. But the human brain involves a lot of infoldings of the cortex. So we have what we call sulci, which are the valleys, and the gyri, which are these outer areas, and the mouse brain is actually smooth. If there's there are diseases in which the human brain becomes smooth. They're called lissencephalic diseases, and in those cases, the person does not have anything you would recognize as human behavior, really. So basically, the folding of the cortex is very important to human behavior. The other thing is, if you look at the mouse brain, the olfactory bulb, or the part that mediates smell, is very large and prominent in the mouse. It's actually not even visible in this view of the human brain, and the parts that are prominent for us are the frontal cortex and the occipital cortex. The frontal cortex, which mediates our higher cognition, and the occipital cortex, which mediates our vision. So we're very different animals in a sense than mouse, and the mouse brain can only offer certain clues about what's going on in the human brain. The other thing is, if you look at how the cortex is structured, so this has to do with looking at the top of the cortex to the bottom of the cortex, and we actually divide the cortex into layers. Many of the genes that are expressed in the lower layers of mouse cortex are expressed in the higher layers of human cortex. And so the higher layers really mediate that the communications between brain regions. So what this means is that when we're studying circuits in the mouse, we might not be looking at the same circuits. The circuits probably don't function in the same way that they function in human. In mouse, these the genes that are involved might be involved more in signaling down into the center of the brain rather than between brain regions. And then the last thing is, if you just want to consider all the possible model systems we use in neuroscience, including cell cultures, you can see that there are kind of three things that we're concerned about: human relevance, mechanistic insight, which is to say, how well does the system teach us what's going on at the molecular level, and then throughput, which is how fast can we study things in that system. And that's important when you're considering things like screens for interventions or molecular screens for possible drug therapies. And what you can see here is human postmortem tissue is the most human relevant. However, lower animals and mouse models offer a lot of mechanistic insight, and stem cells, iPSCs, induced pluripotent stem cells, and neural progenitor stem cells offer high throughput, meaning we can do a lot of experiments in a given day. So when we're considering what's important to inform our understanding of the genetics of autism, we believe that human postmortem tissue is ideal to study because although model systems are great for studying mechanisms, human tissue is the best for understanding what's relevant in human. So I'm going to talk a little bit about molecular convergence of autism risk, and I'll talk about it at the RNA level and the histone acetylation level, and then we'll go into some future directions. So first, let's start with the RNA level. So as I mentioned in the previous slide, the RNA, there's this molecular hierarchy that occurs in every single cell, and that starts at the DNA. It goes up to this chromatin, which is how the proteins modify the DNA, and this chromatin is regulated by multiple other factors. And then it goes to transcription, which is the RNA, and that's also regulated. And the transcripts are translated to protein, and that level is also regulated, and these proteins ultimately mediate signaling. So when we measure RNA and transcription, the real value of it is that it sits in the middle of all of this action, and it can inform our understanding of what's going on at the protein level, at the chromatin level, and what's going on with the DNA. And I'll show you how some of that is related in the next few slides. So the reason, another reason we really like to study RNA is that it tells us a lot about the neurobiological hierarchy. So, for example, first let's just look at this panel. So this is the gene SMARC22, and in fact, I probably have the luxury of making it larger here. Didn't work so well. So this is the gene SMARC22, and what you can see here is this gene is highly expressed during fetal development, but it decreases in expression as you go across time into through infancy, childhood, and adulthood. You can also see that it's highly expressed in certain brain regions. These CTX ones are cortical brain regions, and less expressed in other regions. And it's very highly expressed in cell types such as astrocytes and neurons, but less so in microglia, which are the inflammatory mediators in the brain. Another gene like MET here, you can see that it increases in expression across brain development, highest in adulthood. And in fact, its predominant expression is in these cortical regions. It's barely expressed at all in non-cortical regions, and it's mostly expressed in neurons. And if we look at yet another gene, OLIG1, you can see that that exhibits a similar pattern across brain development as MET, but it's predominantly expressed in white matter, and it's in a cell called oligodendrocytes, almost exclusively in oligodendrocytes. And oligodendrocytes are the cells that mediate the insulating of wires in the brain, you could say. So axons that go between brain regions form are basically how you have long distance connections in the brain, and oligodendrocytes provide what's called myelin, which sheaths these axons, and that forms your white matter, so all of that makes sense. But the important thing to recognize from this is that when we measure the transcriptome, we're measuring about 50,000 genes. This is just 3 of them, and you can see how they have these very rich patterns across brain development, across brain regions, and across cell types. And we can learn a lot about the neurobiological hierarchies by just studying gene expression. So how do we study gene expression? We use a method called RNA sequencing. What that means is we take every RNA molecule, so there's going to be millions and millions of RNA molecules and billions of cells in a given sample. And we convert them to the complementary DNA strand using some biochemistry, and then we slice up all those strands into pieces, and we get small DNA fragments. And these DNA fragments are basically representing amounts of RNA. And when we sequence them, we can align them to the genome. So we can take the letters that are in each fragment and see where they pile up in the human genome sequence. And when we look at a given gene, for example, this is a gene called parvalbumin, we can see that these are cDNA molecules, which really represent RNA molecules, pile up in certain regions, and this allows us to quantify gene expression levels. And in the work I'm going to show you, we predominantly look at 3 brain regions. We look at frontal cortex, because as I mentioned previously, the prefrontal cortex is what mediates higher level human cognition, and this is one of the areas that is affected in autism. We look at the temporal cortex because frontotemporal connections are very important for language development and language function. And multiple neuroimaging studies support the role of the frontal cortex and the temporal cortex in autism. But also, we know from many other studies in like dyslexia, for example, that the temporal cortex is important along with the frontal cortex. And then you have the cerebellum. So the cerebellum is classically, from a neurological perspective, understood to mediate things like balance. But recent studies really support a role in autism and its role in sort of mediating the connections between many of these other brain regions and how they talk to each other. The interesting thing about the cerebellum is that it's also very different from the other 2 brain regions that we're studying. So the 2 cortical regions basically look the same under the microscope, very small differences, whereas the cerebellum looks dramatically different. So it also served as a sort of as a comparator brain region in our analysis. So the first thing I want to show you is one of the very initial experiments we did was to see, can we replicate ASD-associated gene signatures? So what we did is we looked at individuals with autism, and we looked at controls, and we looked at those 2 cortical regions, the frontal cortex and the temporal cortex. And in our previous work, which was published in 2011, we looked at 16 individuals with autism and 16 individuals who did not have autism. And we compared gene expression in them. In our new work, we had an approximately similar number of age-matched samples. And what we wanted to ask is what we saw in this study back in 2011 the same as what we're seeing in our new samples? When we compare the 2, so you can see here on the y-axis is the results from the old study. So each of these points represents the change in gene expression. So it's a log 2-fold change. So 1 here means the gene changed twice as much, and a -1 means the gene is half as much in autism compared to controls. And so here, these are all the results under a certain statistical threshold from our 2011 study, and these are all the results for the same genes, but in entirely new samples with a different method in our new study. What we found is that this R² value is 0.6. What that means is that you can basically draw a line through this and say that there's a high predictive value that if a gene changed in this old study, it also changed in this study. And this is really remarkable because in autism genetics, meaning when we're just looking at the DNA level, we need 10 to 1,000 times this sample size to see the same type of reproducibility and signal. So when you need far fewer samples to see reproducibility, that suggests that there's some level of convergent molecular biology occurring in autism. The other thing we did is we looked at gene expression in, as I mentioned, in cortex, which is what I showed you in the previous slide, but we also looked at it in cerebellum. And this histogram here, basically this peak here says that there's a signal in cortex. And when you compare to cerebellum, the signal is much weaker. And in fact, under the statistical thresholds that are considered rigorous, it's completely lacking the signal in cerebellum. So that means that the molecular changes from whatever is the cause of autism in these individuals and the subsequent development in these individuals, the molecular changes are much more salient in cortex than they are in cerebellum. And this was informative to us because it suggests that there are certain cell types or certain biochemical properties associated with cortical brain regions that are more influenced in autism. We were able to take the changes we saw in cortex. As I mentioned, in cerebellum, there weren't enough changes under a rigorous statistical threshold to really look at, but we were able to take the changes in cortex and see what pathways are affected. And for the pathways that were decreased in autism, they included functions such as the activity of transporters of molecules in cells and neurons, the channel activity, meaning how neurons signal, calcium binding, transport of certain molecules, and transmission of synaptic activity, basically. And for what was increased in autism, we saw that these were pathways largely associated with immune signaling, positive regulation of signaling related to inflammation. So the next question we asked is, what cell types are these pathways influencing? And we found that for the cell types that are, for the genes that are decreased in autism, the cell types are predominantly neurons and those oligodendrocytes, so those cells that are mediating the connections between brain regions. But most importantly, the strongest signal is in neurons. So you can see that this value is very significant. Lower values are significant when we look at statistical significance. And then for what's increased in autism, we saw it was astrocyte signaling and microglial signaling. And microglia are the cell types that mediate inflammation. So the pathways and cell type data are agreeing with each other and saying that basically what's decreased in autism is the ability of these synapses, or kind of the expression level of the genes that mediate synaptic function, and what's increased is the expression of genes that mediate inflammation. And we had seen this previously in our work, and this is basically validating what we had previously seen. And we know that there's also challenges in postmortem tissue gene expression studies. So for example, what we're interested in is frontal cortex, let's say, and what's happening in autism, but other factors that influence gene expression too. Those include the sex of the individual, male or female, the age of the individual, whether or not they were taking medications, whether or not they have seizures, the amount of environmental exposure, that's a playground, that the individual had. So all these can influence gene expression. So we have certain statistical models that try to tease out the signal related to autism versus other factors. And what we found is that we were indeed picking out a signal. The genes we found to be differential expressed are predominantly related to autism diagnosis, not many of the other factors that we were able to measure that could confuse our interpretation of the data. That includes sex, age, region, the quality of RNA, where the brains came from, how long the person had been deceased prior to us receiving the brain sample, the pH of the brain sample, and several other factors. Then the other observation we had kind of made a preliminary version of initially that we wanted to follow up on was the difference between frontal cortex and temporal cortex. So if you compare these two brain regions in controls, you see that there's 551 genes different between them. And you can see that there's this red blotch of genes here. This represents about half of that number of genes, which is up in the frontal cortex, and another red blotch, which is up in the temporal cortex. And this pattern really distinguishes these two brain regions. So that's what we see in controls. When we look in autism, what we see is that there's only 51 genes different between the two regions, and that this pattern is no longer evident. So you can see that we have this distinct red and distinct blue here. It's no longer a distinct red and a distinct blue here. And we were able to do a pathway analysis on the genes. So there are a few genes that actually gain patterning in autism, which I won't go into too much, but the difference between the genes that have patterning in controls and have patterning in autism is 523. And the pathways were really interesting to us from a molecular biology standpoint because things like Wnt receptor signaling, G protein signaling are all things related to the development and differentiation of neurons during brain development. So we wanted to investigate what was possibly driving this loss of cortical patterning in autism. And to do that, we used a method called transcription factor binding site enrichment. So if you look here, what you can see is for the 523 genes we had, we looked at 1,000 base pairs upstream, and we looked at the DNA there, and we asked, do you see a similar sequence of DNA before all of these genes? If you do, that might suggest that the same molecule is regulating all of these genes. And one of the motifs we saw was for a molecule called SOX5. The reason that was interesting is because SOX5 has been previously implicated in regulating neuronal migration, differentiation, and projection of cortical neurons, and it's also been previously implicated in the generation of distinct neuronal subtypes. And when we looked between the temporal cortex and frontal cortex in autism compared to controls, what we can see in controls is that SOX5 is consistently highly expressed in frontal cortex and lower expressed in temporal cortex. So these lines represent the same individual. So in any given individual, SOX5 is almost always higher in frontal cortex compared to temporal cortex. But in autism, that's the case in a few of those individuals, but in others, SOX5 is expressed the same in frontal cortex and temporal cortex, and in other cases, it's actually expressed in the opposite pattern. And so there's this alteration of cortical patterning in SOX5. So what we predicted is that SOX5 is important to the generation of the pattern that differentiates frontal cortex and temporal cortex, and that this pattern is lost in autism. And to validate this, we did an experiment where we took neural progenitor cells, which represent an earlier time point of brain development, and we increased the levels of SOX5. When we increase the levels of SOX5, that decreases the levels of other genes that are involved in cell proliferation and cell movement. These are pathways involved in very early brain development. And increasing SOX5 increases the levels of genes involved in synaptic development and neuron projection, so neurons connecting to each other. This is consistent with a known role of SOX5, and we're seeing this in human neurons for the first time. And we also saw that of the genes that lost their pattern between the two regions, the genes we saw in our experiment highly overlap. So basically you were 2 times as likely to see overlapping genes than you were with background. And when we look within the genes that are affected by SOX5 in the cortical patterning set, we see that most of them have a minus 0.42 fold change, meaning they're decreased in expression. Whereas when we look at genes that aren't regulated by SOX5, they don't really have a change. It's a minus 0.02. So this is really important because SOX5 is known to be a transcriptional repressor. It means that when SOX5 levels go up, it promotes brain development. And so what's happening in this case is that the normal pattern of SOX5 going up in frontal cortex doesn't seem to be occurring in autism. And that's affecting the pattern of these other genes that are downstream of SOX5. But this is important because as we try to understand the function of many different genes in autism, we want to be able to know, are these genes controlled by the same molecular switches, like SOX5 in this case, or are they all in different individual independent pathways, and will we have to understand them all differently? The other thing we looked at was genome-wide splicing changes in autism. So as I had shown you this molecule parvalbumin before and how the RNA sequencing reads pile up in different areas, one thing we can do is we can just sum up all of these reads and say this is the expression level of parvalbumin. But the other thing we can do is we can see how the structure of the parvalbumin molecule and the transcript changes. One way we can do that is, you can see here, this is the parvalbumin gene, but there's 3 actual different genes, or rather 3 different, what we call isoforms of the gene, or 3 different transcripts that come off of the same area of the genome. And we can understand this transcript structure. I won't go into this too much right now, but the main thing I want you to know is that this is independent of looking at gene expression levels. It's looking at transcript structure. What we were able to see is that there was a cortex-specific change in transcript structure in autism and not in cerebellum. And that when we looked at the cell types where this was happening, it was mostly happening in neurons, not in astrocytes, oligodendrocytes, or microglia. So there's a neuron-specific change in transcript structure at autism. And further work we did, which I won't show you here, identified some of the molecular regulators of those, and also showed that a change in neuronal excitation in autism, or in a cell, induces these same splicing changes. So basically, when there's hyperactivity, for example, seizure-like activity in cells, this splicing pattern is induced, and that's the autism-associated splicing pattern. The other thing we were able to do with the postmortem tissue at hand was look at a specific subtype of autism called duplication 15q syndrome. And the prediction we had is if you have the same genetic kit, you're going to have more homogeneous gene expression. So there's variability across the autism spectrum in gene expression we were observing. But based on having a defined mutation, we thought we might see a more clean signature. And then the other question we had was, will the RNA expression changes in duplication 15q syndrome represent what we see in the other autism cases where we don't know the genetic cause or the environmental cause or whatever it might be? So duplication 15q syndrome is a disorder where there is an extra piece of this 15th chromosome, and it results in developmental delay, learning and cognitive disabilities, seizures, and dysmorphias. About 90% of individuals who have the duplication have autism, and this is really remarkable because this is a much higher rate than, for example, the individuals who have Fragile X mutations. So this, what we would say in the terms of genetics, is that duplication 15q syndrome is highly penetrant for the phenotype of autism. So we looked within the 15q region, which is where this duplication occurs, and we looked at gene expression. So just to remind you again, a log2 fold change of 1 is a doubling of gene expression, and blue here is going to be duplication 15q, and red is going to be the idiopathic autism. So in the 15q region, we see that there's a doubling of gene expression in individuals who have the duplication 15q mutation. Not in all genes, but in the vast majority of genes. When we look at the individuals who have autism of an unexplained cause, idiopathic autism, we see that there's almost no change across this region. So what this is showing you is that individuals with duplication 15q mutations have a specific doubling of gene expression within that region of the genome. And this is expected because there's twice as much genetic material there. But what happens throughout the rest of the genome? So what we did is we plotted what's going on in the duplication 15q individuals on the y-axis here, and what's going on in the idiopathic autism cases on the x-axis. And what we found is that you can basically draw a line through there and it's highly correlated, both for the gene expression changes and for the splicing changes. So this is remarkable because we have a defined genetic subtype of autism, and we have a gene expression signature that is across the entire genome that's associated with it. And that gene expression signature is very identical to the expression signature we see in individuals who don't have a defined cause of autism. So what this demonstrates is that we have, regardless of the sort of cause of autism, it looks like many individuals with autism end up with the same genetic signature in their brain. The same transcriptomic signature in their brain, I should say. So the conclusions from this part of the talk are that there are shared molecular changes in autism cortex despite different underlying causes, that newly characterized dysregulation of cortical patterning, splicing, and molecular regulators is further informing our understandings of the molecular biology of autism, and that duplication 15q syndrome recapitulates idiopathic autism at the transcriptomic level. So moving along, this second part has to do with the histone modifications and acetylation. I'll go through this a little quicker. So basically, this is looking at a different level of regulation. It's going to look at how the DNA is modified. What we're measuring here are what we call H3K27AC marks. These are marks for enhancers. Enhancers are regions of the genome where transcription factors, so proteins, can bind and activate or repress transcription. And these epigenetic marks basically tell us where enhancers are. So in order to measure them, we do an experiment called ChIP-seq. So we have an antibody which binds to the histone mark. It basically lets us pull down that part of the DNA. We cut off the DNA flanking the regions, and we only keep what's wrapping around where this histone mark is. And then we map it to the genome when we get peaks, the same as we got peaks. Well, it's the same as the sort of pileup we saw with RNA sequencing, but we call these histone peaks. And these peaks actually most of the time sit outside of genes, and many of them are known to, the ones that are functionally characterized are known to either regulate the gene next to it or sometimes even genes very far away. And that has to do with the three-dimensional structure of the genome and how things are wrapped up in three-dimensional space. So I won't go too much into that. But the important thing is these enhancer marks tell us where, what parts of the DNA are open for transcription or closed for transcription. So we compared autism and control, and you can see this is kind of what our data would look like. So you'd see peaks, for example, in autism, but not in control, or vice versa. And what we found is that most of the signal was confined to the prefrontal cortex and temporal cortex. So you can see that they have about 1,200 upregulated peaks and 1,200 downregulated peaks shared between them, and 1,000 or so of their own. And then there's very little going on in the cerebellum. So this is very similar to what we saw at the transcriptional level. And we also looked at duplication 15q syndrome and saw something similar to what we saw at the transcriptomic level, which is that if we look at what's going on in duplication 15q and we look at what's going on in idiopathic autism, there's a high correlation, meaning you can basically draw a line between it and what's going on in duplication 15q for a given gene, or in this case a histone peak, predicts what's going on in idiopathic autism. We also looked at pathways affected in by both what's up and down in these regions, and what we see is that the differential histone peaks support similar pathways as RNA-seq, but there are also distinct pathways, for example, related to transcriptional regulation and histone regulation, and related to more specific signaling pathways in some cases. So this highlights the value of looking at different levels of molecular biology, because just because something is affected at a transcriptomic level doesn't mean it's going to be affected at the epigenetic histone modification level, and vice versa. So for example, a given DNA change might influence enhancer function in a given brain region, but not do the same thing for transcript function in that same brain region. Then the other observation we made is that, and this has kind of been noted from other experiments in other systems too, is that histone acetylation is associated with transcription. So if you look at the expression level of a gene, if it changes in autism, there's a decent chance that there is a histone peak around it, which also changes in autism. And so this made us wonder, can we use information about the histone peaks to identify transcription factors that might be regulating the RNA level? And so what we did there is we did that same transcription factor search. Instead of looking 1,000 base pairs upstream of a gene, we looked in H3K27AC peaks. And that identified a whole slew of transcription factors, including two that we'd seen in our own work before. I just mentioned the SOX family and SOX5 in particular, but then there's also the MEF family, which I won't go into too much right now, but I can talk more about, which MEF is related to neuronal excitability. And SOX, as I mentioned, those are really important in coordinating neuronal development and particularly cortical layering. So from this part of the talk, I wanted to highlight that histone modifications of DNA support and extend the observations seen by transcriptomics, and that molecular pathway, cell type, and brain region levels, as measured by epigenetics, show convergence despite different putative causes of autism. So what's going on in the future? So the first thing I wanted to highlight here is this. So we understand regulatory networks underlying brain development best in mouse currently. So based on what we know from mouse, there's a period of neuronal proliferation, and then these neurons differentiate, some going to lower cortical regions, layers, lower cortical layers, layers 5 and 6, and others going to upper cortical layers, layers 1 through 4. And we have a moderate understanding of the transcription factors that mediate the various steps in this process. We have a much looser understanding in humans, and part of that is because we can't manipulate molecules in humans the same way we do in mice, but another part is that with mice, you effectively have an infinite amount of tissue available. In humans, we have a very limited amount of tissue. What people have been able to do in blood tissue and in certain other tissue types like cancer tissue that are much more available is they've been able to take an integrative genomics approach where they'll measure multiple levels of biology in the same individual, the genetics, the transcripts, the protein, the metabolites, even the microbiome. And you can use statistical models to basically infer some of these same regulatory networks. So you can go from a genetic architecture, which is what we're getting towards, a very good genetic architecture in autism, to a molecular architecture, but currently we really lack that level of resolution in autism, but also in our general understanding of brain disease. The other advance that is really improving our ability to do biology in the brain and understand disorders like autism in particular is single-cell RNA sequencing and chromatin methods. So in this case, what we do is we can take a postmortem brain, we can take out the nuclei, we can separate all these nuclei out into their individual as individual nuclei, and we can just sequence what's in every single cell and then look at the entire population of cells. What's neat about this is that instead of looking at a bulk of tissue, you're looking at thousands and thousands of cells in each sample. And that identifies not just the difference between neurons and glia, like I was showing you, but it can identify many, many different subtypes of excitatory and inhibitory neurons. And that's going to be really important for understanding of disorders like autism in particular, because we know that there are subtle shifts in excitation and inhibition across different brain regions. And although we can get a course level view of that with postmortem transcriptomics in the way I showed you, the postmortem transcriptomics of the future will have much better resolution. So I want to thank the families involved with the research and donating tissue. As I mentioned, the genetic studies of autism can use blood, and most of our understanding of the genetics of autism is based on whole blood that's been collected. And mechanistic studies have made a lot of progress too, using model systems, human cell culture systems. But so far, we're relatively not as advanced in our understanding of what's going on in the actual human brain in autism because we lack postmortem tissue. Just as a comparison, there's a lot of postmortem tissue available for neurodegenerative diseases like Alzheimer's and Parkinson's. And there's many reasons for that. Part of it is that most of those individuals are towards the end of their life when they are deceased. But because of the availability of tissue, there's a much better understanding of what's actually going on in the human brain in those diseases. So ultimately, molecular genomics methods will improve, but tissue will always be an issue. And looking forward, as technology improves, we hope that our data and the inferences we've made will continue to inform experiments, and our data from postmortem tissue is available for all researchers to use and build upon through PsychEncode and the NIH. Just as a side note, this is a picture of my wife and I here at the Pasadena walk for autism, and we were at the It Takes Brains booth just talking to, I think we saw a good 50 to 150 people. I did it 2 years. One of them was much busier than the other for our booth. It was really great to just talk to people about their thoughts about donating brains, and I don't know, we just had very good discussions. Of course, I'd like to thank our group leader, Dan Geschwin, and many of the lab members who were involved in our research. And then the histone modification work was largely done by our collaborators at the Genome Institute of Singapore, Sean Prabhakar's lab. PsychEncode has been very important for housing our data and for back and forth discussions with other groups. And then the splicing analysis was mediated by our collaborators in Canada and in Europe, so Manuel Arimia and Benjamin Blanco. And we have had funding from Simon's Foundation and the NIH. I'd like to apologize for having to go through things a little fast and not having the animations available. I think the slides were much busier than they would have been otherwise. But I'm happy to go back to anything and discuss answers to any questions that you might have. So we can open up the floor now.
We did get, thank you. First of all, thank you, Neil. That was amazing. You went through it. These are all really difficult kind of concepts for people who aren't familiar with them to get their hands around. So thank you so much. We did get a couple questions. So one of them is, as we learn more about autism genetics and associated brain pathology, do you think that that will change the diagnosis of ASD? In other words, will individuals be given a genetic-based or neurobiological-based diagnosis instead of a behavioral diagnosis? What do you think?
That's a really good question, yeah. I think that there will be a place for that in the long run, but there's also the option to have both in the long run. So you could have the behavioral diagnosis to facilitate, for example, environmental interventions like early therapy and having a sort of enriched environments and individualized development programs throughout education. But you can have the more molecular diagnosis to guide perhaps treatment of comorbidities like seizures. So I think that's definitely a possibility. One of the barriers to that right now is having good markers for autism in a living individual. So we can have genetics, and as I mentioned here, the genetics can be very heterogeneous, meaning that there could be 100 different genes, and maybe there's going to be hundreds of different genes, and maybe 20 of them are contributing in a particular child with autism. So what might be more helpful in directing that child's treatment in the long term might be something we see in brain imaging, for example, but we don't have enough brain imaging yet. We don't have enough information at the brain region and circuit level in humans with autism yet to know how we can use that data.
That makes sense. We have another few that have come in. I know we're going slightly over, but there's a few that have come in around how this informs treatment. So the question is, how does this translate to treatments? Does this mean that the field is moving towards more biological interventions? What about the findings on the immunology and the inflammation response? So I think just answering how these can be used to treatment, it always comes up.
Yeah, there's two major areas that our work highlighted in particular. So one is inflammation and the other is excitation inhibition. So with the inflammation end, I didn't show you this data, but it's in our paper that came out December of last year. But one thing we see when we look across ages in autism is we see that the inflammation signal is really there early on in autism. And so that suggests that if you can somehow reset the microglia, or you can, microglia being the cells that mediate the inflammation, or if you can get that inflammation under control early on, you might be able to have a good biological intervention. The other place our work highlights is the excitation inhibition aspect of things. So, for example, I mentioned that if you basically overexcite cells, you get the same splicing pattern changes we see in autism. And so one of the possible interventions in the future would be to look at perhaps sub-threshold seizure-like activity in autism. So it doesn't manifest entirely like a seizure, but it is basically hyperactive if you were to do a brain scan, like an EEG. And if we could get that sort of activity to be under control or normalized in some way, then it would also, our work suggests that it would also have positive benefits for how patterning develops and how the different brain regions end up in autism. A third area that I should mention also is that we highlight this similarity between duplication 15q syndrome and idiopathic autism. The reason that's important is because it suggests that studying certain types of autism, like duplication 15q, and there's several other types, those subtypes of autism might serve as more homogeneous disorders where we can better understand interventions first and then move them to the broader autism spectrum.
Thank you. That was really informative. I know people are leaving, but we do have one question. One more question that I think you may spend a lot of time answering, but that's okay. So can you speculate what might be causing the differences between SOX5 expression between ASD and control brains? Because there's background variation. Oh, sorry. Could it be due to background variation, or could there be non-genetic factors involved?
Yeah, so our work doesn't give us an idea about that. It doesn't give us a clear answer to that, but I can give you a couple of observations. So SOX5 is a transcription factor that's expressed throughout brain development, as you saw here. And there's multiple ways that SOX5 could be affected and result in the pattern that we see here. So one of the ways, which is supported by other studies out there, is that there are known rare mutations in SOX5 itself. And those have been linked to intellectual disability, but not autism. So that suggests that if you were to actually disrupt the function of SOX5 itself directly, you would have very severe consequences for brain development. What we found in our work, and this is part of the paper that was published in December of this past year, is that we see a signal in like smaller genetic changes, not rare strong genetic changes, but these genetic polymorphisms in the targets of SOX5. And so that suggests that where SOX5 typically binds during brain development, genetic variation, the background genetic variation in the population might be affecting those binding sites. And that might be what increases the risk for autism in the general population in a SOX5 mediated fashion. Now, the critical thing is that SOX5 is one of several other transcription factors that are involved in brain development, and we weren't able to fully interrogate all the transcription factors because you know, we're only looking at a couple of brain regions. We're looking at only adult, either we're looking at older brains basically than what would be ideal. So it's there's a good possibility that if you had younger brains, let's say you had single cell data, you would identify not just SOX5 but several other transcription factors as mediating this cortical patterning effect. And yeah, so basically to summarize, we believe that SOX5 is definitely one of the mediators of what is seen in autism, but what causes the change in patterning could be something in SOX5 itself, and the evidence suggests that that's a very severe consequence, or in the targets of SOX5. And that's what our work currently supports for the broader autism spectrum. And that is what's related to the background genetic variation.
So Neil, I want to thank you again. We're a little over time. I know people have to jump off. There was a couple more questions that I think I'm going to forward to you individually, and then I'll get back to them. But thank you so much. It is always so important to hear about not only why brain tissue research is important, but what the research is showing with it and how it's contributing to autism. And thank you so much. This has been an amazing, unexpectedly amazing, because I always anticipate these genetic talks to be boring and dry, but this one really wasn't. So thank you so much, and thank you everyone for joining us today. As I mentioned on the chat, this will be posted early next week on autismbrainnet.org. There will be a link to a YouTube site. So thank you so much for participating, and we also are having another one of these about, again, a little bit more about the genetic determinants of autism using chromosome 15 and epigenetics, given by Janine LaSalle. So next Friday, I guess, at, again, 1 p.m. this time, Eastern. Thank you guys so much, and if you have any questions, just email me. You have my email address. Thanks, everybody.